So I am inviting the second speaker on stage, and this is Professor Roberto Pini from the University of Florence, who has very successful in creating European networks, and also has been the director of a research institute in the CNR in Florence. Roberto, thank you.
So I have a presentation, but I will try to go very fast through this. If I can get presentation mode. Okay, so I am at the university, but I'm mainly, I was the director of the Institute of Applied Physics in Florence, which is an institute of about 150 people dealing with photonics also, and with medical application of photonics. And so some of my presentation will be about that, but I'm also, on the other side, I am a member of the Pediatric Ethical Committee of Tuscany, so I have to validate, to evaluate clinical protocols, and you can imagine that when pediatric patients are regarded, it's very important, and it's very critical. Okay. In my institute, there are many groups. One of this group was my old group. I had, when I started to deal with paperwork, I had around 20 people working with me. They were young people, and now they are no more young. And there are no more one group, there are 4 groups now. And they're dealing with biophotonics. Biophotonics is photonics for medical application, but also for biological application. So we have something which is related also to plants, to agriculture, to agri-food, where we use fluorescence methods to study plant physiology and product quality. Diagnostics is used for recognition of biomarkers in particular for neurodegenerative diseases. Other methods like advanced microscopy, we use multiphoton microscopy and other types of microscopies. Nanomedicine, well, this is the name of the group. I mean, we do biophotonics. We would like to do something related to nanomedicine in the future, but this is our hope. What happens? Why going back? Okay. We also do therapy, not only diagnostics. So we use lasers. I was originally a laser man. 40 years ago, I was build-up laser with my hand, and we tried at that time to find a useful application in many fields, like the medical field. About using laser, I've done many things about laser surgery, but not talking about that here. We work on laser-assisted repair of biological tissue, the so-called laser welding or laser soldering of tissue. We have developed devices for photocoagulation, abrasion, and wound healing. Now they are used to treat diabetic wounds, and they work very well, even if I don't believe the physics which is behind, but they work. And in the last 10 years, we were also involved in nanomedicine. In particular, we are using nanoparticles that we develop for tumor theranostics. I'll say some more words. And we are also working on drug release using some specific devices that can be implanted, and that can be most of these applications are driven by light. So I'm a friend of light. I'm not afraid of light. Because we would like to use light as something like a trigger to start some particular procedure. What you see here with the dashed line are the applications which are in the preclinical phase, which means that they are applied in animal models or in patients for preclinical trials, while these applications are already in the clinical phase. They are used in real patients. Okay, my background is not very far from yours. I mean, I use light, lasers, and chromophore or constant agents, something that absorbs light, to perform hyperthermia, which is a controlled heating of tissue, which can be applied also for tissue bonding of tissue. And in particular, you can try to bond connective tissue like collagen, elastin, and so on. And this had application. I'm reporting here just the main application in ophthalmology, microvascular surgery, neurosurgery, and so on. So it's very important that we want to validate a method like this, that you start with materials and drugs that are already validated for the clinical use. So we started with indocyanine green, which is absorbed in the near infrared region. For instance, one of the main peak of indocyanine green is around 800 nanometers, where you can get a diode laser to excite. And the diode laser has a deep penetration depth into tissue. So whenever you stain the wound that you want to repair with indocyanine green, you have a very selective absorption and release of heat exactly in the wound. And you can control the release of heat in order to have a denaturation, control the denaturation of the collagen. You don't want to destroy the fibrils, but you want just to break the bond between the fibrils in order to speed up the healing process. And this was applied from 2000 to 2010, 2015, in about 500 patients to assist the transplant of the cornea, where there is a lot of collagen. And this was done to substitute the conventional suturing. Usually, when they transplant the cornea, they used to apply what they call a continuous suturing, which takes a long time and use a lot of suture materials. So you can imagine that if you can skip this material, you have lesser inflammation because you can minimize the foreign body reaction. And also, if you bond, you have a stable astigmatism at the time of surgery that doesn't change during the healing time, and the faster healing time. We did the same thing also for arteries, and this was applied at the Neurosurgical Institute of the Catholic University in Rome for the bypass of the carotid artery, because in that case you need to have an occlusion of the artery for a very short time, because otherwise you may have a damage to the brain. So it's important if you can replace standard suturing with laser welding, laser soldering. And this was done also to repair or to produce anastomosis in microvascular structures. So then we moved from constant agent to nanoparticles. And in particular, we like to work with nanoparticles that could be activated by electromagnetic fields or light. That's why we were using magnetic nanoparticles or metallic nanoparticles that can absorb light in the visible or in the near infrared, depending on the shape of the nanoparticle. So we develop in our labs many types of nanoparticles, in particular gold nanorods. And here you can see very compressed the work that we have done in more than 10 years, developing nanoparticles for targeted hyperthermia of tumor cell, of tumor mass. And we performed 3 different strategies. One was to use this gold nanoparticle absorbing near infrared light with onboard a specific cancer biomarker. This was applied, for instance, anti-125 antibody to target specific cancer like ovarian cancer. Another strategy was to target the environment where the tumor is developing, because typically when you have a tumor, cells are in a hypoxic state because there is no lack of oxygen. And when in this condition, typically cells have a process in which they produce some particular species like CA9 that can be targeted by specific sulfamidic agent that can be on board of these nanoparticles. And then you shine the light, and then you can perform hyperthermia. And the last strategy was to use cellular vehicles, in particular macrophages or lymphocyte T killers, to reach the tumor. But before we could take them from the patient, loaded with gold nanorods, and then get back to the patient to reach the tumor. And so this was a strategy that was brought up to the animal models, but not to patients. And with this experience in nanoplasmonic particles, we also develop diagnostics and platform for to monitor biological proteins based on surface-enhanced Raman spectroscopy, which is a spectroscopy based on Raman, but when the biological protein is in contact with the metallic surface, you have an enhancement of a Raman signal that can be many order of magnitudes, 10 to the 14 even. And with these nanoparticles, we had different solutions, nanowires, nanocubes, nano other materials. We developed platform in order to detect some particular biomarkers, for instance, beta amyloids oligomers, which has been recognized as the biomarkers of many neurological diseases like Alzheimer's disease, or to produce a wearable chip sensor for monitor metabolites in sweat. And this is just a very fast sketch about the application on Alzheimer's disease. We produced this platform, and then we studied the spectra that you can obtain with this particular surface-enhanced Raman spectroscopy platform. And then we performed artificial intelligence calculation in order to clusterize the people which had some probability to exhibit Alzheimer's disease in the future with other people which are not affected of the same disease. And this is a story which is very, very rapidly synthesized, but it was just to let you understand that sometimes you have to use tools that has to be validated. And this is a story involving artificial intelligence now in health and medicine, because the applications are growing very rapidly. Whenever you have a large amount of data, you need to elaborate them and try to find the biomarkers which are important for some detections, from some diagnostics. But you have to consider that to some extent, artificial intelligence and machine learning methods, and especially those involving biophotonics, must move from the lab to clinics to gain the approval as medical devices. So in my experience with medical devices, where we have to validate and certify this system according to the European regulation. And this is the ethics committee. Okay, this is just a court of judges, but this is felt like this. I mean, from the scientists, from the researcher that has to validate the clinics, and so there are some regulation that you have to account. And the last one is the Artificial Intelligence Act, which was approved a few days ago. And so you have to consider that still there are no exact regulation that are related to the use of these tools on patients. But each country and each region in our case has to give the right indication in order to use properly these applications. So let me just skip to the last part of my talk. I want just to present some opportunities that you can find, if I can get the general, okay, here. I was last week at the Photonics Partnership annual meeting in Brussels, and this is the European platform for photonics, and there is a work package which is devoted to health. But nevertheless, you may know that, let me go to presentation. that now photonics is no more one of the 6 key enabling technologies for industrial innovation, but after Horizon Europe is part of digital technologies together with micro and nanoelectronics, and also artificial intelligence is another digital technology. So whenever you're looking for a call or a work plan, you cannot find photonics on its own. I mean, you can find photonics with other things. For instance, now it's very popular to have this chip join undertaking, and a lot of money, a lot of billion are on it, because we have to get rid of this innovation, this dependence from other countries. When I was there in Brussels, they presented also the 8 calls of the innovative health initiatives, where you can find probably some interesting application. But just a few words, I'm concluding about a hub, a digital innovation hub, which is called Photon Hub Europe, which started in 2021, soon after the pandemic, but it is extended up to April 2026, and it is a very good occasion to promote innovation with small and medium enterprises related to photonics and also to medical application of photonics. I am the coordinator of the CNR in Italy for that. The coordinator of the whole project is Professor Hugo Tiempont from Brussels. And the idea is to create something like a one-stop shop providing services to companies. And the role of a research group is to bring the companies there. There are 4 pillars, as you may know, for the Digital Innovation Hub. Training, supporting to find investment, to create an innovation ecosystem. But one of the most important one is to produce some prototypes. Test before invest means that you have to demonstrate that your technology is really working. Okay, so let's keep these. There are many companies, many technologies like medical optics and so on. But the opportunity is here that depending on the TRL of the innovation the company want to promote, you can get a support. I mean, not a large amount of money, but this is sufficient to start, up to 100,000 euros, but with a support which is 75% of money, and the rest is cash from the company, from the group. And depending on the TRL, this goes down to 50%, and when you are close to the manufacturing and production, it goes down to 0, but the network can help to diffuse the information about the innovation. So there is this opportunity. It goes up to the 2026. So whenever you know a company, the condition which is strict is that only the projects that are cross-border can be supported. Cross-border means that you had to put together a small and medium company with a lab which is in another country of Europe. Okay, this is the condition. But whenever you find this opportunity, for instance, you want to develop some specific chip or so, and you can go to this lab or to this company, then you can get also a very strong percentage of support depending on the TRL of your innovation. So this is the website where you can get all the information, and if you want specific information, you can ask to me and write down at this address. Thank you.